Now, what we discussed in previous parts of these discussions, that action and its explanation cannot coexist with each other, right? The action and its explanation cannot coexist with each other. For example, I saw this hand, the notion of this object as a hand, the very objectification of this hand via different sorts of superimposition or discontinuity is not in this hand. The name hand is not in this hand. So where all those informations are getting stored, that we are interested to look at, right? That's one thing. So action cannot have its explanation within itself. The explanation has to be somewhere else. And to understand it more clearly, we also discussed there is absolute continuity. That is, that is our, what we think as our body is in continuity with all other actions. Our body is in continuum, or is in continuity with all the other discrete objects that we objectified. Everything is in continuity, the whole universe. And if that is the case, then where all these explanations are getting stored? Because actions or action cannot coexist with the explanation, right? Or information. Information is basically our explanation. Then we have to go through the process of observation, how the information is getting stored, because some people say that the brain is the storage of the information. No, brain is the objectified action that is connected with the whole universe, and it's in continuity, and it's an action, and action cannot coexist with the explanation. And an action doesn't have the desire to explain anything, because the action cannot observe itself. The observer can look at the action, but the action cannot look at the action itself. Make this very, very clear. Don't bring any prejudice or any borrowed arguments that you heard during some other masters or teachers. They maybe use them for different context, but go through your observation process. Develop your own observation skills. Try to understand what we are going through. We have to go through together, side by side. It's not like I'm giving you some beliefs and you are believing it, all those things. So information has to be stored in somewhere else, which is not the action itself. That's the crucial thing to observe or understand. So as we just discussed, information is nothing but our explanations. And how we try to explain things, we try to explain things by first we develop an explanation for a particular action, let's say rising the sun, or how the birth of a tree occurs, or how we walk, what's the mechanism of walking. So these are all actions. So we projected our explanations on each action, like this is walking, this is the sun, this is the setting of sun, this is the rising of sun, this is the earth. So all those informations or explanations has to be somewhere else than the action itself. Make that very, very clear, okay? So now the and now how we verify that this is a correct explanation or this is a wrong explanation? We try to reproduce or try to verify that in an act of experiment. So what is experiment is? Experiment is creating similar conditions that will create a similar kind of action. The action that we saw, it's gone. You cannot reproduce it because for that particular action, the whole universe is acting together. That's that's gone, and that was never separate. So what was there, it is also right now. So it's not gone, but we have the illusion that it's gone. So it's very complicated. So very calmly focus on what is continuity, and then try to understand what we call as experiments in laboratory or whatever possible way we uh, try to prove that my theory is correct, that theory is wrong. The main point is, if something will happen, it will happen. It doesn't mean that your explanation is correct or false. The sun will rise and it will set. That's a fact. It doesn't matter what explanation you give. And in all the attempts that you took throughout your lifetime to explain that unexplainable action is a waste of time. I mean, in absolute sense, it's a waste of time, but in another sense, those explanations can be very helpful to know that they are absolute waste of time. If we don't wasted any time, we won't be able to know that we wasted our time, right? This is also very critical point to understand. If you don't have any falsity or false item, you won't be able to falsify that. And without the ability to falsify anything, without the ability to observe anything, you won't be able to know the observer, or you won't be able to know the truth, which is falsifying everything. You see, there is only truth, there is only observer, but the illusion of falsity, or the illusion of other the object or the action, that is there. If that is not there, we won't be able to get a clue of who the observer is, or who, what is truth is, what is false, all these things, you know. So that's very important. So we know that what is our explanations. And another point is, what will happen in lower level of reality. It doesn't give a damn of what explanation you have. In another higher level of reality, there is nothing happening at all, because everything is continuous. We will go slowly from step 1 to step 2, step 3, 4, 5, 6, 7, maybe not more than that, right? Okay, we will see how many steps are there. And also there is going down steps are also there. Steps and steps, all the way. It's going to the up, it's going to the down. We will see how the steps are going up and going down. Okay, so storage of information. This is the most important or most interesting thing that we want to explore. Why it is interesting? Because we now know that action and its meaning cannot coexist with each other. Make that very clear. Action and its meaning cannot coexist with each other. So then comes the obvious question. So now I hope for all of you it's very clear that brain is not the storage of the information, because brain is also the part of the continuity of this whole universe, right? We are going to look at the very biology or chemistry of the cell, how everything is happening. So let's start. So this is a cell. Don't be scared. This will be very short, I promise. This is not a biology class. So this is a cell. We know that inside the cell there is all sorts of chemicals. One of the most important thing is the nucleus, which has a chromosome. This is the chromosome, and chromosome is nothing but the DNA. It's rolled, or it's coiled in a particular shape. Based on that shape, we call some chromosomes at X, some chromosome at Y. Now, DNA is composed of different base pairs like A, T, G, C, you know. And what is ATGC? ATGC are chemicals like adenine, guanine, thymine, cytosine, uracil. Uracil is not present in DNA, but it is present in RNA. So how the RNA and DNA is formed, or it's getting formed, that to know that we need to know this ribose sugar. This ribose sugar is connected with the nitrogens that are empty, or that is having the hydrogen as a substituent. So they will be connected in this particular position, and it will form. This is not RNA, by the way. This is the nucleotide. This species is nucleotide. And you see this difference between these two sugars. One is having no hydroxyl group is this in this particular position. Another is having hydroxyl group. So the one that doesn't have hydroxyl group called deoxyribose sugar. The one that has is just called the ribose sugar. So when it is deoxyribose, the DNA term is coined. So DNA means deoxyribonucleic acid. And this whole double helix structure is basically the interaction, the hydrogen bonding interaction between the A and T. This is A, this is T, and G and C. So you see here A and T are interacting, G and C is interacting. This interaction is always between A and T, G and C, because of the hydrogen bonding interactions. The people who knows little bit about chemistry, they will be knowing what I'm trying to say. And in DNA, this is the nucleotide that will be used. In RNA, the nucleotide will be same, only there will be an extra hydroxyl group. That's it. And also the DNA is double helix, the RNA is single helix. And this is the phosphate that also has to be attached with this. So based on the number of phosphates that this particular nucleoside is having, we call it mononucleotide, dinucleotide, trinucleotide, right? And there is also different amino acids based on different codons. So codons are the three base pairs. So for example, this ATAC or ATG corresponds to the methionine. There you have GGC or CCG. These are corresponding to the proline, like that. And this is the amino acid, the general structure of amino acid. You has to have a carboxylic acid. There has to be a NH2 group, and you have different substituents on this particular position. So when it is CH3 or methyl, you have alanine. When there is nothing, it's glycine. When there is something else, it's called something else. We have specific numbers of amino acids. So those amino acids are combined with each other via this bond. This bond is called the peptide bond, right? And through this peptide linkage, the macromolecule that is formed is called a protein. So this is a protein. So protein has all sorts of activities in the living cell, and these are basically macromolecules. They are basically the instrument which is producing all sorts of small molecules which are either getting metabolized or getting produced for different sorts of actions, right? So I know what I'm saying. So just to show you, this is a publication that I got during my postdoc tenure in Princeton. So I know what I'm talking. So let's go to the next slide. So this is the enzyme. When the enzyme has certain capability of producing small molecules through chemical reaction, we call it enzyme. Enzyme has active site. So this is the active site of the enzyme. This is the cofactor. You see this yellow color chemical or compound. This is the catalyst which will remain as it is, but it will be helping in performing the reaction that the enzyme is supposed to catalyze, right? So all enzymes are catalysts. They remain always same, but they catalyze different reactions. And what are the things the enzymes are doing? So these are the macromolecules. Enzymes are macromolecules. When there is surrounding interactions, let's say I am interacting with the surrounding, whatever surrounding environment I am. Let's say I ate something which is, let's say, a banana or something that has macromolecules like cellulose or different polymers of sugar that needs to break down for metabolism. So that job will be done by the macromolecules, another macromolecules, which are called enzymes. So they will interact with the small molecule, with that food, let's say, what we took from the surrounding, and they will interact with it to produce small molecules, right? So taking any food is interaction with the surrounding, right? And then these small molecules will create different actions, like when the polymer will be the glucose polymer will be, let's say, maltose or sucrose. When they will be break down into smaller constituents like fructose or glucose, they will create different actions like producing energy for walking, running, or speaking. And we objectify each actions, right? And these actions doesn't have any meaning. They're just actions, but we are projecting our meaning that this is digestion, this is eating, this is enzyme, all those biology or chemistry that we just discussed in the previous slides. Those things doesn't have the meaning. Even right now, the slide doesn't have any meaning. These are just shapes, right? This surrounding, this interaction, these are just painting drawn on the canvas. But they are actions. But who is projecting the meaning that this word surrounding or interactions or small molecules, macromolecules, who are projecting these meanings? Those words or those actions doesn't these words are colors, right? We perceive them as colors in the black canvas, but who gave them the meaning? They didn't give the meaning. First of all, we created this shape which is called the alphabets of a script called Roman to make a language or to utter a language called English. Those are all our creations, right? But what is creation? We saw this writing down action, and we gave the meaning that this is This action is called small. This action is called molecules. This action is called an arrow, like that. Those interpretations, these interpretations are projected by us. They are not present in the action. Make this very, very clear what I'm trying to say. Actions doesn't have the meanings or interpretations with them. We interpret them as we wish. That is how the models of science gets improved, right? From very old days to the middle age, then to the different centuries, and right now it is getting refined and refined and refined, right? So our interpretations is getting changed. We are interpreting them, right? The interpretations are not there. We are interpreting them. Based on what? Based on actions. After observing an action, we are interpreting them, that this is that, this is that. The interpretations are not present in the action. We are projecting the interpretations. Make this very, very clear. And all those chemistry, the structures, the enzymes, the active site, those interpretations are our interpretations. It's not there in the cells, in uh, the very background that you are looking at. They are all shapes and names, but the interpretation of each shape, each name is projected by us. That this word will be meaning this thing, that word will be meaning that other thing. That meaning is projected by us, right? That's a crucial thing. And any interpretations or explanations or informations, and informations cannot stay with the action, because the action is not interpreting itself. It's the observer who is it's the observer with the desire who is basically willing to interpret or willing to explain all the things, because he wants to explain, or he wants to enjoy the same explanation again and again, or maybe some other enjoyments like that. So the desire has to be there. And the most noticeable thing is the actions and its interpretations has to be different. For us who are right now here, I mean, who are going through this journey, right now it is very clear that they are different. They cannot be coexisting with each other, right? There is other examples like this. Small molecules, let's say we have different hormones, right? Different hormones and different chemicals that we take within us, like drug molecules which act as medicines. So they also interact with different macromolecules. It can be enzyme, it can be protein. After interacting, it will block the native activity of the enzyme or the protein, which will inhibit some actions. So the macromolecule or the enzyme were supposed to produce any action. That action maybe we are not willing to perceive. We are, because we already gave that action a name. That action can be fever, that action can be headache or something, and we want to get rid of that action. Sorry, we want to get rid of that emotion. The emotion is not in the action. We want to get rid of that emotion by stopping that action, right? So uh, when we take any small molecules as a drug, or as a addiction, or as any other supplement, they are basically blocking or enhancing the features of small molecules or macromolecules, sorry, macromolecules or enzymes, because these macromolecules are basically the uh, moderator of the small molecules. They control the production or the metabolism of the small molecules. These are very, very basic things in biology. And these, everything is objectified by us. So after objectifying each actions as separate or as discontinuous action from the whole continuity. We interpreted them as metabolism, we interpreted them as interaction between a small molecule and an enzyme, analgesic interactions like that. And those interpretations are not present in the molecule or within the atom or within anything. Those interpretations are where that we are gonna look at, right? So the same mechanism that we just saw for a cell, let's say normal tissue, skin cell or blood cell, whatever, but that is also true for the brain cells, right? I mean, the most useful cell of the central nervous system is the neuron or the nerve cells, right? So if someone argues that the whole eye, the neuron, the brain, they are basically interacting or they are basically acting as a knowing instrument or as the perceiver of the outside color or whatever interactions we observe by them, then first of all we need to see that whether these cells, these eye, the neuron, the brain, are they engaged in any sort of action with the surrounding. If they are engaged in any sort of action with the surrounding, that means their action itself, right? Because there is continuity, no one can deny it. If they are interacting with the action itself, that means their action itself. Because the only way you can interact with an action is through an action. I mean, this hand is holding this pen and I'm just shaking like this. The whole act is an action. The pen is action, the hand is action, the shaking is action, because action can only interact with action, right? Action can only interact with action. So if, and also that, I mean, the we are perceiving all actions, right, through our sense organs. So if I can perceive my hand, that means I how I am perceiving. I am seeing the color, the touch sensations, the smell, the taste, etc. Right? And those are all actions that I am perceiving. And whoever is perceiving is different from the action, right? Because the very act of perceiving is projection of different explanation. That this is my hand, first projection. It has a color, second projection. It has a smell, another projection. I can hold something. The concept of holding something is another projection, projection after projection. And those projection or explanation cannot be either by my hand, which is an action, right? Because I'm perceiving it and it's also engaged in other actions. And that is also connected with my brain. Brain is also interacting with my throughout the body, right? The whole thing is action, and action cannot give meaning. And then an interesting thing comes, that how we speak. I mean, the only way we can give meaning is I'm not gonna say the only way, because we can also write, and by writing we can convey our meaning, but that's secondary, right? The preliminary way, or the preliminary connection, how we express our emotion, is sound. By uttering our urge to explain something, or by uttering our urge to say a name, to exhibit the meaning that we want to convey by uttering that name. The most initial state of expressing the desire is the sound, right? That we will come. I'm just saying the thing, how it is, how the observation is going to be. So that's an interesting thing. So what we saw that the brain, the neuron, the eye, they are all part of a whole continuum of action. They are action itself, and they cannot project any meaning, or they cannot superimpose. That it's the same for atoms. So this is a hydrogen atom, and the structure will be same for other atoms. Sorry, similar for other atoms. They will have a nucleus. They will have the different number of electrons. So the atom or the subatomic particle, they are the very constituents of what we superimpose as matter, right? Matter. But they are just objects, and whatever is object is action, because we can perceive them, and whatever we can perceive is only action. We cannot perceive the object as it is, or the object itself. We can only perceive the action, the color, smell, touch, taste, and sound, right? And whatever we can perceive cannot project meaning on them. The actions cannot project meaning on itself, or right now my hand is not able to project any meaning. The only preliminary way of projecting meaning is sound. And how the sound is connected and how it is, we are going to see, we are going to look at it. But right now just see that the action cannot project itself the meaning. The action cannot project the meaning on itself, right? Make that very, very clear. Okay, so there is the atom, and then there is the cell. The cell of the neurons will be, the neurons will have the same chromosomes and everything. uh, they will have different chemical environments, they will have different shapes, but the basic constituents, that is the atom or molecules, organic molecules, they will be same, similar nature. The DNA will be same, the ATGCU. U is uracil, which is exclusively for RNA. We have different sugars, phosphates. So so all these are interpretations, right? And these interpretations cannot be with the action itself. The action cannot interpret itself, because only the observer can interpret the action or can project the meaning based on the desire, right? And an action cannot have any desire. Our brain cells are working constantly. They are active, interacting with the surroundings. They are taking information, analyzing it, and then giving other informations that this is how you should do. Like when I touch a hot plate, I am supposed to take it back immediately. And those are the sensory and the motor nerves that we have in our body. They are constantly doing those things. Right. So those are the actions that is happening. They cannot project any meaning on what they are doing. Contemplate on it. This is very absolute clear fact. Okay. So right now what we just saw, that all these small molecule, large molecule business that we see inside a cell, their objectification, that this is an enzyme, this is catalyzing a reaction, producing a small molecule which is again engaged in different actions like producing heat, etc. So those are all objectifications of discrete events that we think as discrete, and those discretion or those act of objectification or objectifying is done based on the sense perceptions that we have. If we try to analyze by different instruments, it depends on the sense perceptions of the instrument. I mean, if the instrument is capable of only visually take the image or something, the instrument doesn't have any sense organs. I'm just trying to explain how the instrument is interacting with the object or with the action that we want to see. And based on that interaction, which is also an action, we are interpreting that this is this, this has to be an atom, this has to be a molecule. We will come to the different phenomenons of quantum mechanics or classical mechanics. We will see that all of them are superimposed or explained by us. They are not what is happening, we don't know. We are projecting our meanings on based on those actions, and we are objectifying each actions, and that is actually we can use it in a very very exciting way to know us for our self knowledge. Okay, so these are all interpretations, and these interpretations cannot be stored in brain, that we also saw. Why? Because brain is action, since it is interacting with other actions. Only an action can interact with other actions, right? And the observer has to be different than the action. Um, and based on the desire to explain, the observer project different meanings on the actions that it observes. And the interesting thing then comes is where all the information is getting stored then. And this is the first point, interesting point, or the most uh, exciting question. The second question is, or the second fact is, rather, that the superimposed imaginations or any explanations cannot stay with the reality or the higher level of reality of the continuity of existence, right? Because the whole continuous existence is actions or action that we perceive by our sense organs, and whatever is perceived is action, and whatever is perceived cannot project meaning on it. The entity who is perceiving or observing has the only ability to throw meaning on what he is observing, right? So the observer and the thing being observed is has to be different based on what criteria? Based on the criteria of superimposition. The observer only has the capability of superimposing, not the things being observed, right? We are not going straight to that higher level of reality. Just go step by step, make it very very clear in the stage where we are right now, right? Okay. And then another fact that this diagram can be helpful for us just to remind that all actions are observable, but they are indescribable. We describe them as different descriptions. Those are called informations or explanations or imaginations, and they has to be stored in somewhere else because they cannot coexist with the action that we perceive. And that storage is also an object to the observer, right? So that storage has to be in between of the action that we perceive. Then there is the storage of all those informations, and the observer is also also there, who is having access to all those things. So these are also some interesting facts to consider. And there is also the continuity. So we will see that whether all these different looking phenomena, they are at all they are at all continuous, or they are discontinuous as we are observing right now. So don't put or don't bring any assumptions, just go through the facts. We will be able to actually observe the higher levels of reality if we follow the facts from the lower level of reality. Any jump or any attempt to know the higher by skipping the lower states will be disastrous. So let's go step by step, right?